Hello. Hello. Hello, everyone. My name is Snehlata and I'm a PhD student and a PMR fellow in the Department of Biological Sciences and Bioengineering at IIT Kanpur. Today, we will continue with the next lecture of the chapter Photosynthesis in Higher Plants. So till now, we were studying about the photosynthesis. We covered the experiments, the early experiments that were done to prove that what components are important for the photosynthesis. Then we saw that in what type of reactions happens in photosynthesis and what are the pigments that are required for it. In the previous lecture, we saw that the pigments are basically divided based on their presence in different level of plants, such as in simpler plants like algae, in higher plants and bacteria. Today, we will see the classification of pigments that are present in photosynthesis on the basis of their functions. Okay. So, on the basis of the function, the pigments are divided into two types. Okay. So, on the basis of the function, the pigments are divided into two types. Now, what are these types? So, the first type is called as reaction center. So, the pigment of the first type is known as reaction center. And the second type is known as antenna. Okay. So, on the basis of function, these are two types of pigments. Now, let's see about the reaction center. So, in the reaction center, the pigment that is present is known as chief pigment or the primary pigment. Okay. So, the pigment in the reaction center is the chief pigment. Now, why do we call it chief pigment? We will see in the upcoming slides. But for now, just think it as the most important pigment. Okay. Now, what does this chief pigment do? So this chief pigment, it absorbs light. It absorbs light from antenna. Okay. Because why it absorbs light? Because in last lecture, we saw that the pigments undergo excitation by absorbing light, right? So they excite their electron, and when this electron come back to the ground state, they release some kind of energy, either in the form of light or heat. And in meanwhile, it also gives like the light of different wavelength that is responsible for different colors, right? So this chief pigment, from where it absorbs light, so it absorbs light from antenna. Now, what is the third important thing about the reaction center? The important thing is that it also contains enzymes. So the reaction center pigments, it contains enzymes, okay? Contains enzymes, which means that it is capable of catalyzing the reactions, right? And we know that either it's a plant or animal, these reactions are responsible for everything that we do. Or we can simply say it is responsible for our survival. So this chief pigment contains enzymes that catalyzes the certain biochemical reactions. Now, what is the example of chief pigment or the reaction center? So the example is chlorophyll A. In higher plants, the example is chlorophyll A. And in the previous lecture, we have seen the structure and the formula of chlorophyll A, right? Okay. Now, let's move to the antenna. So antennas are known as accessory pigments. Okay. So these are known as accessory pigments. So it means the antenna comprises of a group of pigments, and these pigments together called as accessory pigments. Why? Because these accessory pigments, they absorb light directly from the sun. Okay. They absorb light from the sun, and then these pigments transfer the light, or you can say energy, because light is a form of energy, right? So it absorbs the energy from the sun, and then it transfers that energy to the reaction center. Okay. So suppose this is your accessory pigment, and this is your accessory pigment. So once it will absorb light, then this light will be transferred to your reaction center. Okay. It will be transferred to the reaction center. Okay. So because of this, because it is helping the reaction center, these pigments are known as accessory pigments. So let's write. So accessory pigments are what? These are the pigment that absorbs light and transfer it to reaction center. Okay, transfer it to reaction center. Okay, because of this reason, these are called as accessory pigments. Now, the light that it absorbs, it is of a specific wavelength. Okay, so it's not like it can absorb light of any wavelength. So the light that this antenna absorbs, these are of a specific wavelength, or you can say a fixed wavelength. Okay. Now, these antenna are also known as shield pigments. These are also known as shield pigments. Now, why these are known as shield pigments? Because when was in last lecture, we saw that there are certain situations when the light intensity can be very high. And in that case, there is high chance that it can damage the reaction center if it gets exposed to the light directly. Okay. So what does the shield pigments do? These shield pigments, they prevent the plant, or you can say reaction center. They prevent the reaction center. They prevent the reaction center from photooxidative damage. From photooxidative damage. Now, why I'm saying so, photooxidative damage? Because in the case of high light intensity, when it reaches the reaction center, it can lead to the formation of oxidative radicals. Okay, so these radicals, these are very toxic and they can damage the reaction center. And because it is happening due to high light intensity, hence it is called as photooxidative. Okay, so this antenna also acts as a shield pigment that prevent the reaction center from photooxidative damage. Now, what is the source of light or energy for this antenna? So these antenna can absorb light. These can absorb light either directly from sun, okay, either directly from sun or neighboring antennas, or they can also absorb light from neighboring antennas. Okay, so this I'll explain with an example, but for now, just remember that these antenna can absorb light either directly from sun or from neighboring antenna. Okay, and what are the examples of accessory pigment? So out of many pigments that we studied, one of the example is chlorophyll B, that I'm representing by CHLB, and carotenoids. Okay, and carotenoids. And these carotenoids especially, they act as a shield pigment. Okay, they act as a shield pigment. Now you can think that what is there in this carotenoids or the antenna that they do not undergo photooxidative damage, because if the light intensity is very high, it might be possible that these antenna itself undergo photooxidative damage. But what happens that these antenna, they are comprises of a group of pigments, right? So they comprises of a pigment, something like this. So suppose this is 1, 2, 3. These are all the antennas. These are all your antenna. And here is your reaction center. Okay, this is your reaction center. Now, these antenna, what happens? Suppose this absorbs light. This absorbs light, okay? And this light is a very high intensity. But as it moves, because it is not only one, it comprises of many such pigments, right? So once it absorbs light, what it does, it causes the excitation of the electron, right? The electron undergoes excitation. And when it comes back to the ground state, it transfers its energy to the neighboring antenna, right? So now the energy that is absorbed by or received by this neighboring antenna, it will be less as compared to the first one, right? And in this way, that by the time it reaches to this reaction center, the energy minimizes or it gets attenuated so that it does not affect this reaction center, right? So this is how all these pigments work. So here you can see that say that this what you call, these are your antennas. These are your antenna, and this is your reaction center. Okay, this is your reaction center. So when it absorbs light, it will transfer it like this, and these are of the wavelength. Like this is, suppose if it is of wavelength 500 nm. So next should be of higher wavelength, because we know, and we saw in the previous lecture as well, that pigments always transfer their energy to a higher wavelength, right? And why this happens? Because we know with the formula that E is equal to H by lambda, C by lambda, right? So here energy is inversely proportional to lambda, that is wavelength, right? That is, so this will be H, okay? That is wavelength. So higher the energy means lower the wavelength. If and lower the energy means higher wavelength. Okay. So once it is transferring its energy, it will always be towards the higher wavelength, not the lower wavelength. So the next pigment to which it will transfer energy will always be higher than 500. So say it's 510, 520, 530, likewise. And by the time it reaches the reaction center, it will be say 680. So what we can think, or what we can conclude from here, that reaction center have the highest wavelength, right? Because it is absorbing light from its neighboring antennas, and light or the energy can only be transferred when the reaction center will have the highest wavelength. So we can write that reaction center have highest wavelength. Okay, have highest wavelength. So this is about the pigments, that is reaction center and antenna, based on their functions. Now let's talk about what a photosystem is. So whatever diagram we are seeing here, this together is comprising the photosystem, but we will see in detail like what the photosystem is. Okay. So the photosystem, it comprises of, in simple terms, it comprises of pigments and proteins. Pigments and proteins. Now, these proteins are what? These are enzymes, basically, because we know that enzymes are proteinaceous in nature. Okay. So the photosystem comprises of pigments and proteins. So if we have to divide, so photosystem, I am representing by PS. So it has what? It has first the light harvesting complex. Light harvesting complex. It has light harvesting complex, that is represented by LHC. Okay. Second, the photosystem have electron carriers. Okay. Electron carriers. So what does this electron carriers do? They carry the electron and transfer it from one component to another, right? So, and we know that transfer or lose or gain of electron is known as oxidation and reduction, right? So these electron carriers, what they do? They cause oxidation. They cause oxidation and reduction. Okay. And third thing, what they have enzymes. The photosystems have enzymes. Now, what is light harvesting complex? So light harvesting complex is nothing but this thing. So this, whatever I have drawn here, this is your light harvesting complex. Why? Because here what we are seeing that light is getting absorbed by one of the pigment, then it is sequentially getting transferred to the nearby pigments and finally to the reaction center. So this together, this unit comprises of the light harvesting complex. So this together, this unit you can call it as LHC, or light harvesting complex. Okay. So the photosystem has light harvesting complex. Apart from this, it has electron carriers and enzymes. Now, what are the types of photosynthesis? What are the types of photosystems? So photosystems are of two types. Okay. These are of two types. The first is known as photosystem 1. The first type is known as photosystem 1, or it is also called as PS1. Okay, PS1. And the second type is known as photosystem 2. Second type is known as photosystem 2, known as PS2. Okay. Now, if we talk about the location of this photosystem 1 and photosystem 2, so in the last lecture, what we saw that this photosynthesis, it takes place in the mesophyll cell, and in the mesophyll cell itself, it takes place in the chloroplast, right? So here, what we labeled in the last lecture, that the chloroplast comprises of your thylakoids, grana, and stromal lamellae. Now, in this, the photosynthesis happens somewhere. And this photosynthesis is caused by or is performed by these photosystems. Okay. So, where are these photosystems located? So, the photosystems are located. If we talk about the PS1, so the PS1, it is located on the somewhere on over the this membrane. Okay. So, the photosystem, it is located on the this thylakoid membrane, any membrane if you talk about, except for the chloroplast membrane. So what membrane do we have? We have thylakoid membrane and we have stromal lamellae. The photosystem, it is located on the thylakoid membrane. It is located on the thylakoid membrane and stromal lamellae. Stromal lamellae, right? Because it is the membrane that is exposed. So you can also say that this is representing PS1. So the PS1 is present on the exposed region of the grana, or you can say in the chloroplast, it is present in the exposed region over the membrane. Okay. So the photosystem 1, it is present on the thylakoid membrane and the stromal lamellae. Now, where is photosystem 2 present? So the photosystem 2, it is present in the non-exposed part. Okay. Non-exposed part. Now, if you look into this structure, where is the non-exposed part? So the non-exposed part, although it is present on the membrane, but on the non-exposed part. So it will be somewhere like here, somewhere here, right? So it is present in the inner thylakoid membrane, right? So this is your what? PS2. So PS2 is located into inner thylakoid membrane. That is located on the inner thylakoid membrane. That is present where? To the lumen side, because this region, this is known as lumen. Okay. So it is present in the lumen side. Okay. So we have seen that where the photosystem 1 and photosystem 2 is present. And this photosystem 1 and 2, these are mainly responsible for photosynthesis. Okay. These are responsible for photosynthesis. Now, let's see what is the difference between the photosystem 1 and photosystem 2. So first of all, let's talk about why they are not named as 1 and 2. Okay. So they are named as photosystems. These are named on the basis of their or order of their discovery. Okay. So the photosystems, these are named in the order of their discovery. In the order of their discovery, which means this photosystem 1, it is discovered first. That's why it is called as first. Okay. So discovered first. And photosystem 2, it is discovered second. Okay, right? It is discovered after PS1. And because of after PS1, right? So the photosystem 1 and 2, they are named after in the order of their discovery. Now, in the photosystem 1, what type of reaction center is present? Because why I'm talking about reaction center here? Because the photosystems, what they comprises of? They comprises of this light harvesting complex. And if you go back to the previous slide, the light harvesting complex has the reaction center and antenna, right? So it means that the photosystem will have some reaction center. So the reaction center that is present in photosystem 1, so the reaction center in photosystem 1 is chlorophyll A, and that get excited at 700 nm. Okay, that get excited 700 nm, which means that if you give a wavelength of light of 700, it will undergo maximum excitation. Okay, so the photosystem 1 has a reaction center that excited 700 nm, and because of this reason, this photosystem 1 is also known as PS700. So if you see a name PS700, So it means the photosystem 1. And it is named so because it gets excited at 700 nm. Okay. Now, what kind of reaction center do photosystem 2 have? So the photosystem 2, the reaction center in photosystem 2 is reaction center in photosystem 2, it get excites at 680 nm. Excites at 680 nm. And because of this, it is known as PS 680. Okay. Because of this reason, it is known as PS 680. So the reaction center 1, it's also called as PS 700, and photosystem 2 is called as PS 680. And now we know the reason. Okay. Now, what we also saw that photosystems have light harvesting complex, right? The light harvesting complex consists of reaction center, but apart from reaction center, it comprises of one more thing, and that is antenna, right? So if we have photosystem 1, then what kind of antenna do we have? So the antenna, if we talk about the antenna in the photosystem 1, so they will have wavelength of light less than 700 or higher than 700. So they will have light wavelength of less than, we can have wavelength less than 700 nm, right? Because then only they can transfer their energy to the reaction center, right? Because the reaction center has the highest wavelength, right? And because of this, the antenna will have wavelength less than 700 nm. And in the case of photosystem 2, antenna have wavelength less than 680 nm, because here the reaction center is of 680. So the wavelength of the antenna in the case of photosystem 2 will be of 680 nm. Okay. Now, we have seen in the previous slide also the location. The photosystem 2, it is present in the non-oppressed part, right? Non-oppressed means that is exposed. So it is present in non-oppressed part, or you can say, or exposed part. Exposed part. And we know what that part is. That is your thylakoid membrane and stromal lamellae. Okay. And the photosystem 2, it is present in, the photosystem 2 is present in non-exposed part, right? It is present in non-exposed part. And we know where the non-exposed part is. That is your inner membrane of thylakoid, or what can lumen side of thylakoid. Lumen side of thylakoid. Okay. Okay. Now, okay. So now one important thing to note is that both of this photosystem 1 and photosystem 2, this I am mentioning here, because what we are seeing that the photosystem 1 has a reaction center of 700 nm and the reaction center of photosystem 2 is 680 nm. It means that both of this photosystem can get activated if you give a light of less than 680 nm. Okay. If you give a light higher than 700 nm, none of them will be activated. So both PS1 and PS2 get activated by wavelength that is less than 680 nm. Okay, less than 680 nm. So this is the important point. Now, suppose if you give a condition like where lambda is 550 nm. If you give a wavelength of light that is of 550 nm, then what part of the photosystem 1 and what part of the photosystem 2 will be activated? So at 550 nm, you have here antenna. The antenna will be activated, right? Antenna active, because this will be activated at 700 or slightly less than that. But at 550 nm, the antenna, in both the cases, antenna will be active. Antenna will be active. Now, if you give a light of say 700 nm, so will the photosystem 1 be active? Yes, because the photosystem 1 can get excited till 700 nm. So here the reaction center of the photosystem will be active. reaction center will be active, right? And here, in the same case, if lambda of 700 nm is given, so because its maximum excitation can take place at 680 nm, now if you give a light of wavelength 700 nm, its reaction center will not be active. So the reaction center is inactive, right? Okay, so these are certain scenarios by which you can understand that in what situation photosystem 1 and photosystem 2 will be active. The important point is if you give a light of wavelength less than 680 nm, both the photosystem 1 and 2 will be active. But if you give a light of wavelength 700 nm, only photosystem 1 will be active. Okay, and now the question is, how do we know that the plant chloroplast comprise of two photosystems, the photosystem 1 and photosystem 2. So, in order to know this, in early experiment, there was one experiment that is known as immersion enhancement effect. Okay, so this experiment was performed. It was performed to prove that plants. Why this experiment was performed? So it was performed to prove that plants have two photosystems. Okay, have two photosystems. And the experiment was known as immersion enhancement effect because it was performed by immersion. Okay, so scientist was Immersion. Immersion was the scientist. Okay. Now, what he did, the Immersion, he used chlorella. Chlorella as model organism. Model organism is the organism on which he performed the experiment. So he used chlorella. Okay. So he performed the experiment on the chlorella, and the light source that he used. So the light source that he used was monochromatic light source. Now, what is monochromatic light source? Monochromatic light source means it at one time, or you can say they are different lights. Okay, all are of monochromatic. So they will emit a light of a specific wavelength. Because in your house we have torch or say bulb, LED. So they do not give a light of a specific wavelength. They comprises of all the visible wavelength light. Okay, but the monochromatic light source, they have light of single wavelength. So these source have light of single wavelength. Okay, and hence they are called as monochromatic light source. Now, so this Emerson, he performed a series of experiments. Okay, so first experiment that he performed, performed a series of experiments, out of which in the first experiment, what he did, in the first set of experiment, he gave he gave a light of say a wavelength 500 nm, and then he looked for the effect. Okay, then he checked the photosynthesis. Then he give the light of say 550 nm. Then he give the light of 600 nm. And likewise, when he gave light of 680 nm, when he gave light of 680 nm wavelength, what he observed that there was increase in the rate of photosynthesis. He observed increase in the rate of photosynthesis. Okay. So so he observed an increase in the rate of photosynthesis. Now, why this increase happened? Because now we know, we have studied about the photosystems, and these photosystems are responsible for the photosynthesis. So when he gave the light of 680 nm, one of the photosystem gets activated, right? And we know what photosystem gets activated at 680 nm, that was PS2. Okay, so because of this reason, he observed increase in the rate of photosynthesis. Okay, now when he gradually increased the wavelength, the second experiment, he say move from 680 to 690 or 685. So when he gradually increased the wavelength to greater than 680, say, greater than 680, what he observed? He observed there is a drop in the photosynthesis. Okay, he observed drop in photosynthesis. Now he thought he one might think that what happened, why it gets dropped, right? So there was drop in the photosynthesis, and he called this effect as red drop effect. He called it as red drop effect. Okay, he called it as red drop effect. Okay, now what he did, he performed the third set of experiment. He again performed the third set of experiment where he gave a light of wavelength 700 nm, light of 700 nm wavelength. Now again he observed that there was increase in the photosynthesis. He observed increase in the photosynthesis. Increase in the photosynthesis. And now we know why this happens, because at this time, when the light of 700 nm was given, there was activation of what? PS1, photosystem 1, right? And that leads to the photosynthesis. Now, then fourth, he thought, now let's combine both this 680 and 700 nm and let's see what happens. So in this, he used two light source, one of the wavelength 680 nm and another of the wavelength 700 nm. So he gave combined 680 nm plus 700 nm. So there was one light source that gave 680 nm, then another that gave 700 nm, and then he observed a sharp increase in photosynthesis. Observed sharp increase in photosynthesis. Okay, he observed sharp increase in photosynthesis by 25%. By 25%. So what was his observation? That when he gave the combined two lights, one of 680 nm, another of 700 nm, there was sharp increase in photosynthesis by 25%. So from here, and this effect was known as, because there is enhancement in the photosynthesis, it is known as immersion enhancement effect. It is known as immersion enhancement effect. And from this, he concluded what? That the plants consist of two photosystems. One that is getting activated at 680 nm and another that is getting activated at 700 nm. Okay, so this is the immersion enhancement experiment that proves that plants have two photosystems. Okay. Okay, so this is about the basic of the what you can say a background on which now we will see that what actually happens in the photosynthesis. Okay, so the photosynthesis has, as we have seen previously, it has two types of reactions, right? The first one is light reaction, and the first one is light reaction. So now we'll start with the light reaction. Okay, so in the light reaction, we'll start with the electron transport chain. So what is electron transport chain? So it comprises of electron carriers. So electron transport chain is what? It comprises of electron carriers. Okay, it comprises of electron carriers. Now, where is the electron transport chain located? So it is located in the thylakoid membrane. So just like this, where the PS1 is located, similarly, the electron transport chain will be located somewhere here. Okay, so the electron transport chain, it is located in thylakoid membrane. Okay, thylakoid membrane. Now, so what do we have? What I said, that it comprises of electron carriers. So it's not like there is only one electron carrier. It comprises of series of electron carriers. So this is one electron carrier, this is another, this is third, this is fourth, and they transfer the electron. One, this is one, then it transfers its electron to the next one. It transfers the electron to the X, and it transfers the electron to next one. So likewise, this happens. Now, if someone asks that what is the condition or what requirement is there or you can say under what situation the electron will be get transferred from one carrier to another. Okay, so it is done on the basis of the redox potential of that carrier. Okay, so electrons are transported or transferred based on redox potential of the carriers. Redox potential of the carriers. By carriers, what I mean? By carriers, I mean electron carriers. Okay. So electrons are transferred based on the redox potential of the carriers. Okay. Now, what is redox potential? The question is, what is redox potential? So the redox potential is the ability of a chemical species to either lose or gain electron. Okay. Because we know redox comprises of oxidation and reduction. And oxidation and reduction happens when there is lose or gain of electron. So the redox potential is the ability of a chemical species. It is the ability of a chemical species, okay, to either lose or gain electron. To either lose or gain electron. Okay. So which means if if a chemical species has, you say, high redox potential. So if there is high redox potential of a chemical species, by chemical species, what I mean? I mean the chemical compounds. I'd say we'll come across some here in, say, cytochrome that we'll see. So high redox potential. So what does high redox potential means? It means that that chemical species, that particular element or compound, you can say, it is in oxidized state. Okay, it is in oxidized state. And we know what is oxidized state or what is oxidation. Oxidation is when there is more ability to gain electron, because the oxidation happens when there is loss of electron, right? Now, if a species has lost the electron, it has ability to gain the electron, right? So oxidized state means it has more ability to gain electron, right? So if a chemical species has a higher redox potential, means that it has a more ability to gain electron. And vice versa, if it has low redox potential, means it is in the reduced state. The species is in the reduced state, and hence it has. Okay, so I was talking about the electron transport chain. So in the electron transport chain, what you saw that it comprises of electron carriers. Okay, so the electron carriers, and these carriers are located in the thylakoid membrane, right? So the electron carriers, they are located in the thylakoid membrane, and these electrons are transferred based on the redox potential of the carriers, right? So what I was saying is, suppose there is one chemical species, second chemical species, third and fourth chemical species. So the electron gets transferred from 1 to 2, 2 to 3, 3 to 4. Now the question is, how or under what conditions this electron gets transferred? So the answer was that electrons are transferred based on their redox potential. So electrons are transferred based on the redox potential of the carriers. Okay. Now, what is redox potential? So the redox potential is the ability of a chemical species to either lose or gain electron, right? So it is the ability of a chemical species to either lose or gain electron. Now, if a species has a high redox potential, it means that it is under the oxidation state. Okay, it is under the oxidized state, and it means that it has more ability to gain electron. And if a species has a low redox potential, it means that the species is in the reduced state, and it has a more ability to lose electron. Okay, it has more ability to lose electron. Now, okay, So if I ask that out of these four species, 1, 2, 3, and 4, which has the highest redox potential and which has the lowest redox potential? So the highest redox potential will be the one that has more ability to gain electron, which means the fourth one will have the highest redox potential. It will have the highest redox potential. And because of this, it is gaining electron, right? And the first one, because the one that has more ability to lose electron will have low redox potential, will have low redox potential. Okay. So if we say the first one has low redox potential, first one has low redox potential, and fourth has highest, you can say highest, it has lowest, and the fourth has highest redox potential, highest redox potential. And which means what? Which means that the electrons in the electron transport chain, it simply means that the electrons in the electron transport chain, electrons in electron transport chain I'm representing by ETS. So they move from low redox potential, move from low redox potential, low redox potential to high redox potential, to high redox potential. Okay. And this movement is known as downhill movement. It was discovered by scientist named Hill and is known as downhill movement. Okay. Now, so this is about the electron transport chain. Now, what is what is the use of this electron transport chain? So the question is, what is the use of this electron transport chain? So the use is to create a proton gradient. Okay, so what does proton gradient mean? The difference in the level of protons. Okay, so it is used to create proton gradient. Okay, and that leads to ATP formation. That leads to ATP formation. So why this proton gradient is important? It is important to synthesize ATP. And this electron transport chain, they somehow leads to this creation of proton gradient. And we'll see how they leads to proton gradient creation. Okay. Now, so this is the important, or you can say the utmost importance of the electron transport chain, because it is used to create a proton gradient, and that ultimately leads to ATP formation. Now, what do we understand by ATP formation? ATP formation means that we have ADP, we have phosphate, and then it leads to phosphorylation. So what happens here? Here the phosphorylation of ADP. Phosphorylation means addition of phosphate. So here in the ATP formation, what happens? The ADP is undergo phosphorylation by this phosphate and then lead to the formation of ATP. Okay. So this process of ATP formation is known as phosphorylation. And why it is known as phosphorylation? We just saw. Now, if it is happening during photosynthesis, then this process will be called as photophosphorylation because it is happening in the presence of light. Now, the photophosphorylation. So it is ultimately leading to the ATP formation, right? Now, this photophosphorylation can be of two types. Okay. The first one is known as non-cyclic. And the second one is known as cyclic photophosphorylation. And in the next slide, we will study about the cyclic and non-cyclic photophosphorylations. Okay. So in the first, we'll start with the non-cyclic photophosphorylation. Okay. So what is non-cyclic photophosphorylation? What happens in this? First of all, what is the location of non-cyclic photophosphorylation? So the location is in thylakoid membrane and stroma. It happens in thylakoid membrane and stroma. Now I'll show you how. So if you connect the dots, what we saw that we have photosystems. First of all, we have reaction center and antenna that together comprises of light harvesting complex, right? And this light harvesting complex, what it has, this light harvesting complex is a part of a photosystem. And then we saw what is the location of photosystem. And then we also said that a photosystem is responsible for photosynthesis, right? So these all things we know. Now, if we talk about the location, we are saying that this non-cyclic photophosphorylation is happening in thylakoid membrane and stroma. The question is, how can we see this? So if you remember, I'm drawing a zoom version of a thylakoid. Okay, I'm drawing a zoom version of a thylakoid. So this is your thylakoid, right? Now, what we said that the photosystem 1 and photosystem 2, these are two photosystems that are present, and these are responsible for photosynthesis. So if you remember the photosystem 1, where it was present on the non-exposed part. So photosystem 1 is present here, right? It is present over the membrane. 1 second. So the photosystem 1, yeah. So it is present on the membrane. Okay. So the photosystem 1, it is present on the membrane. This is a photosystem 2. And what we saw, the photosystem, this is your what? These are your photosystem 1, right? And where is the photosystem 2 present? So the photosystem 2 was present in the non-exposed part, or you can say what we said, inner lumen side of the thylakoid membrane. So the photosystem 2, these are present here. These are present here. Okay. So this is your photosystem 1, photosystem 2. So if the photosynthesis will take place, where will it happen? It will happen in the thylakoid membrane. This is your thylakoid membrane, right? And in the stroma, because this is your one thylakoid. The structure that I have drawn is of thylakoids. So this is what? This is thylakoid. This is thylakoid, right? Now this thylakoid is present in a big chloroplast, right? So it is present in a big chloroplast. And here the photosystem 1 is present here. So if it will catalyze some reaction that we'll see, it will go into the stroma, right? So because of this reason, I'm saying that this non-cyclic phosphorylation, it is happening in the thylakoid membrane and the stroma. Okay. Now let's see. Let me erase this and see that how this non-cyclic phosphorylation is taking place. Okay. So the non-cyclic phosphorylation here, it happens. So this is, say, for you. So say this is the inner membrane of the, thylakoid. Okay. This is what? This is inner membrane of thylakoid. Okay. Here you have what? You have your PS2, photosystem 2. So here you have PS2, or it is also known as PS680. Right? We have seen it is known as PS2 or PS680. Now it receives light. Okay. It receives light. Once it receives light, what it will do? Its electron will undergo excitation. Right? Its electron will undergo excitation. Now, who will receive this electron? The electron will be received by the electron transport chains, because they comprises of electron carriers. So the electron that is excited from here, it will be received by electron carrier component known as pheophytin. So it is received by an electron carrier component known as pheophytin. Okay, pheophytin, that is represented by P. Okay, it has received the electron. Now, what will this pheophytin will do? This pheophytin will transfer its electron. It will transfer its electron to another electron carrier that is known as plastoquinone. So it will transfer it to plastoquinone, and it is represented by PQ. Okay. And this is the inner membrane of thylakoid, remember. So this all is happening in the outer membrane. Okay. Or you can say over the membrane. Now this plastoquinone, what it will do? It will transfer the electron to another electron carrier that is known as cytochrome B6F. cytochrome B6F. Okay. Now it will transfer its electron to another electron carrier that is known as plastocyanine. It will transfer to plastocyanine that is represented by PC. Okay. Now this PC will transfer its electron. It will transfer its electron to the photosystem 1. It will transfer it to PS1. It is also known as PS700. Okay. It has transferred. Now, once it will receive the electron, it will transfer the electron. It will get excited and transfer the electron to FES. And this FES, now it transfer the electron. It will transfer the electron to FD, that is ferrodoxin. So it will transfer to ferrodoxin. Okay, that is represented by FD. And then it will lead to the catalysis of a reaction known as NADP plus to NADPH conversion. So here I am representing it. NADP is getting converted to NADPH. Okay, one second. It is getting converted to NADP plus is getting converted to NADPH. Okay, NADPH. Okay, with the help of this electron, right? And not only electron, the NADP also require H plus ion. So the reaction will be NADP plus H plus leads to the NADPH formation. Okay, so what we have seen that the PS2 upon excitation, it gives its electron to electron carrier known as pheophytin, that then transfer it to the plastoquinone, that is PQ, which in turn transfer it to cytochrome B6F, and then it transfer it to plastocyanine, and it ultimately leads to another photosystem, that is PS1. And then from PS1, it gets into FES and then ferrodoxin, and ultimately it gets converted to, or it gets received by NADPH. Now, if you look at the location, so how will you draw this? So we have seen that, this, where is this PS2 is located? It is located in the inner membrane of the thylakoid, and this PS1 is located in the outer membrane, right? So this all reaction is happening between inner and outer membrane. So this is your inner membrane. So their outer membrane will be something like this. Okay, it is located above the outer membrane. So it will be happening something like this. Okay, so if you see, this is your thylakoid. This is inner membrane. This is outer membrane. This is your outer membrane of thylakoid. So this is thylakoid, not chloroplast. Okay. Now it will be located in a big chloroplast. So in the chloroplast, just imagine this region will be what? This will be stroma, right? So this region will be stroma, and this is your thylakoid. So now we can clearly see which of the reaction is happening in the thylakoid membrane and which reaction is happening in the stroma. This much reaction. This much reaction. This reaction is happening. This reaction is happening where? It is happening in the stroma, right? It is happening in the stroma. And the rest of the reaction, these are happening on the thylakoid membrane. Okay. So this is the non-cyclic phosphorylation. Now, what is the important thing to remember from here? So what we are seeing a series of electron transport chain electron carriers, right? So let's talk about this electron carriers. So the first electron carrier that we see is what? Pheophytin. Now, this pheophytin is known as colorless. It is known as colorless with chlorophyll. It is known as colorless chlorophyll. Chlorophyll I am representing by A. Colorless chlorophyll A. Why? Because its structure is similar to chlorophyll, but it do not have any color. Because of this, the pheophytin is known as colorless chlorophyll A. And if you look here, the pheophytin is the primary acceptor of PS680 electron, right? Because the electron that is excited from this photosystem 2, it is directly getting collected by pheophytin, right? So the pheophytin is the primary electron acceptor of PS2. Okay, this is important. Now, let's talk about the second, what you say, electron carrier. And that is what? That is, the second electron carrier is plastoquinone. Okay, so plastoquinone. What do we know about plastoquinone? So plastoquinone is the electron acceptor of pheophytin. This thing I do not need to write. You should remember this chain. Okay. So the plastoquinone, apart from this plastoquinone is a mobile carrier. So it can move. Okay. So it is a mobile carrier. And the important point about the plastoquinone is what? It acts as a hydrogen pump. It acts as a hydrogen ion pump. Okay. So this plastoquinone here, it is present in the membrane, right? So this all is happening in the membrane. So it will plastoquinone, it will be like this much. So the hydrogen ion that is present in the stroma, it will pump this hydrogen ion across here to the lumen, to the lumen. Okay. So this plastoquinone, it acts as a hydrogen ion pump. So it transport, I'm writing this transport hydrogen ion from stroma to lumen. From stroma to lumen. Okay. Now, what is your third electron carrier? It is plastocyanin. The third is although this cytochrome B6, but the important point is about plastocyanin. So plastocyanin, it is also a mobile carrier. So in electron transport chain, we have two mobile carriers. The one is plastoquinone, that is represented by PQ, and another is plastocyanin, that is represented by PC. Okay. Okay. Now, here what we saw, that ultimately the electron is getting accepted by NADP+. So the ultimate acceptor of, or the last acceptor, the ultimate acceptor of electron is NADP+. Because ultimately the electron is getting accepted by NADP+, and it is getting converted into NADPH. Now the question is, what enzyme? Because if some reaction is happening, there must be certain enzymes. Those are catalyzing this reaction. So what enzyme is catalyzing this reaction? So the NADP plus hydrogen ion plus electron is converted into NADPH. Okay. So it is catalyzed by an enzyme known as ferredoxin NADPH reductase. So it is catalyzed by ferredoxin NADPH reductase. Okay. This enzyme is also known as FNR. Ferredoxin NADPH reductase. So this enzyme catalyzes this reaction. And this enzyme is present where? On the stromal side of RENA. Stromal side of thylakoid membrane. Stromal side of thylakoid membrane, right? If you look into the structure, it is present here. It is connected to this PS1 and it is present on the stromal side of the thylakoid membrane. Okay. Okay. So till now, let's end our class here. We will come in the next lecture. We will see that from where this electron is getting replenished, because we are seeing here that the electron is getting used up in the reaction, but from where this electron is coming. So we'll see this. We'll continue in the non-cyclic phosphorylation in the next lecture as well. So for now, that's all. Thank you.